Welcome everyone. Today we are diving into the biological landscape of cell injury, how cells respond to stress, what decides whether they bounce back or die, and how these changes shape diseases. From swelling and fatty change to necrosis and apoptosis, we will uncover processes that form the foundation of pathology. Understanding these mechanisms is not just about exams, it is knowledge that underpins diagnostic, therapies, and future research you might lead. So students, today the title of our topic is Cell Injury, Pathway of Reversible and Irreversible Changes, Damage, Necrosis and Apoptosis. Students, this discussion will cover under the following learning objectives. First, understand causes and mechanisms of cell injury. Second, distinguish reversible and irreversible cell injury. Third, identify patterns of necrosis. Fourth, understand molecular basis of apoptosis. Fifth, relate cell injury to disease and research. Now we will start our topic. Cell injury refers to harmful changes that cells undergo when exposed to internal or external stressors. These can include physical forces like heat or radiation, chemical substances, infections, immune reactions, or imbalances in nutrition and oxygen supply. Depending on the nature and intensity of stress, the damage may be reversible, allowing cell to recover, or irreversible, leading to cell death. When injury overwhelms the cell's repair mechanism, death can occur either by necrosis, a chaotic and damaging process, or by apoptosis, a controlled and programmed pathway. Physical factors may cause injury by disrupting cellular structure, while oxygen or glucose deficiency impairs energy production, especially ATP. Toxins, drugs, or heavy metals may interfere with essential functions by damaging proteins or membranes. Infections also harm cell. Viruses invade and destroy cells from within, whereas bacteria may produce toxins or trigger inflammation. For instance, Clostridium perfringens releases toxins that cause gas gangrene. Key cellular components like the DNA, plasma membrane, and mitochondria are common injury targets. DNA can suffer thousands of lesions daily due to normal metabolism or environmental exposures such as UV radiation. Membrane damage disturbs ion balance, especially calcium levels, which can promote apoptosis. Mitochondrial injury through ATP depletion or membrane disruption also play a critical role in irreversible cell damage. Now we are going to discuss about the types of cell injury. There are two types of cell injury. First is reversible cell injury and second is irreversible cell injury. First we will discuss about reversible cell injury. Acute cell swelling is the cell's immediate reaction to disrupted homeostasis and is considered an early indicator of injury. If the stress is mild and short-lived, the cell can restore balance and return to its normal structure and function, making the injury reversible. Although swelling is a hallmark of reversible damage, it can also appear in the early stages of irreversible injury. A common cause of swelling is hypoxia, which reduces ATP levels and impairs ion pumps on the cell membrane, especially the sodium-potassium ATPase. As a result, the cell loses its ability to regulate volume and takes in excess water. Other factors such as oxidative stress, toxin exposure, membrane damage, or pore-forming complexes can also disrupt membrane integrity and lead to swelling. Because acute swelling occurs in both reversible and early irreversible injury, it is best viewed as a warning sign rather than a definitive outcome. The final fate of the cell recovery or death depends on the severity of damage, duration of exposure, and the cell's capacity to repair. In some cases, even cells on the verge of irreversible damage may recover partially, though they often do not return fully to their original functional state. Now we will discuss about the acute cell swelling. Acute cell swelling is a common and early sign of cellular injury, often referred to as hydropic degeneration. It results from the accumulation of water and sodium inside the cell. When sodium-potassium pumps on the membrane become dysfunctional. If uncorrected, this swelling can progress to cell lysis and death. The term hydropic degeneration is typically used when this condition affects specific cells like liver cells, also called hepatocytes, or renal tubular epithelial cells. In other tissues such as epidermal keratinocytes, the swelling is often called ballooning degeneration. In the central nervous system, also called abbreviated as CNS, the swelling of glial cells, especially astrocytes, is known as cytotoxic edema. Unlike hypertrophy, which involves an increase in cell size due to the growth of organelles or protein content, acute cell swelling is due to water accumulation and reflects a degenerative, not adaptive change. It signifies a disruption in ionic and fluid balance within the cell, not a physiological adaptation. Under light microscopy, cells undergoing acute injury typically appear enlarged, often compressing or shifting nearby structures. Their cytoplasm may lose staining intensity, resulting in a pale or hazy appearance. In some cases, clear vacuoles form within the cytoplasm, usually reflecting the dilation of the endoplasmic reticulum or Golgi apparatus, a change historically referred to as vacuolar degeneration. However, not all affected cells show vacuoles. Instead, their cytoplasm may appear diluted with scattered organelles, a condition sometimes called ballooning degeneration. The nucleus usually remains relatively unchanged in the early stages and tend to retain its normal position and shape for that specific cell type. At the ultrastructural level, typical features of reversible cell injury include formation of cell membrane bulbs, blunting and distortion of microvilli, weakened cell-to-cell junction, swelling and structural changes in mitochondria, dilation of endoplasmic reticulum. Now we are going to discuss about mechanisms of acute cell swelling. In healthy cells, the sodium-potassium ATPase pump actively transport ion across the plasma membrane by using energy from ATP hydrolysis. For each ATP molecule consumed, the pump expels 3 sodium ions from the cell and brings in 2 potassium ions. This activity helps maintain the electrochemical gradient, which is essential for processes such as membrane potential regulation in neurons, heart muscles and skeletal muscles, as well as maintaining intracellular pH balance. Since water moves passively following osmotic gradients, this pump also plays a critical role in regulating cell volume. Acute cell swelling often results from ATP depletion, which impairs pump function such as in hypoxic conditions or from membrane injury, as seen with toxins like carbon tetrachloride. Additionally, compounds like digitalis and ouabain, known as cardiac glycosides, directly inhibit sodium-potassium ATPase, further illustrating the importance of this pump in maintaining cellular integrity. Now we are going to discuss about fatty change, also known as steatosis, a reversible lipid accumulation in injured cells. Fatty change, also known as steatosis, occur when cells, particularly those involved in cell metabolism, are injured and lose the ability to properly process fats. As a result, lipid droplets begin to accumulate in the cytoplasm, forming small vacuoles. While mild lipid buildup may not significantly disrupt cell function, extensive fat accumulation can impair normal cellular activity. In the liver, for instance, the swelling of hepatocytes due to fat overload may press against bile canaliculi, potentially causing cholestasis. This condition is often reversible, depending on the underlying cause and extent of lipid deposition. Other terms for fatty change include fatty degeneration, fatty metamorphosis, and lipid infiltration. Grossly, the affected liver may appear enlarged with a yellow, greasy surface. Microscopically, it is characterized by the presence of clear lipid-filled vacuoles within the cytoplasm of the hepatocytes. Now we are going to discuss about irreversible cell injury. The key causes of acute cell swelling include hypoxia, such as from ischemia, and plasma membrane damage resulting from lipid peroxidation or the formation of lytic pores by mechanisms like the membrane attack complex, also abbreviated as MAC, of the complement system, or bacterial toxins. A cell's reaction to injury is influenced by both the cell type, including its sensitivity or resistance to oxygen, deprivation and membrane damage, and the intensity, nature and duration of the injury. Some cells, such as neurons, cardiac muscle cells, endothelial cells, and renal proximal tubule epithelium, are highly vulnerable to hypoxia. In contrast, fibroblasts, fat cells, and other connective tissue cells tend to be more resistant. Depending on these factors, the response may be reversible, adaptive, or degenerative. However, if the stress is too intense or prolonged, initial swelling can lead to irreversible injury and cell death. Distinguishing between reversible and irreversible cellular changes remain a major area of ongoing research. Now we are going to discuss about necrotic cell death. During normal development and tissue remodeling, the body eliminates cells that are no longer needed. However, when cells experience severe irreversible damage, they undergo cell death as a final protective response. This pathological cell death is the focus of this section and usually presents in one of two distinct forms. One is called necrosis and another is apoptosis. Now we are going to discuss about necrosis. Necrosis is marked by cytoplasmic swelling, also known as oncosis, irreversible damage to the plasma membrane, and disintegration of intracellular organelles, ultimately resulting in cell death. Unlike apoptosis, necrosis often affects clusters or regions of cells and typically triggers an inflammatory response due to the release of intracellular contents into the extracellular matrix, also abbreviated as ECM. The morphological stages of necrosis include pycnosis, which we mean condensation of chromatin and shrinkage of the nucleus; karyorrhexis, fragmentation of the nucleus and chromatin into coarse granules; karyolysis, complete dissolution of the nucleus. Leakage of cytosolic enzymes and proteins through the compromised membrane is a key factor in initiating and amplifying the inflammatory reaction in surrounding tissues. Oncotic cell death, a result of severe cell injury. Oncotic cell death occurs when a cell suffers irreversible damage, often due to lack of oxygen, reduced blood flow, also known as ischemia, or direct damage to the cell membrane. Ischemia is especially harmful because it not only reduces oxygen but also cut off glucose and nutrients while allowing toxic waste products to build up. A key feature of oncotic cell death is cell swelling, caused by the loss of control over fluid and ion balance. This swelling sets it apart from apoptosis, a more controlled form of cell death. Like a reversible injury, oxygen shortage stops normal ATP production by oxidative phosphorylation and forces the cell to use anaerobic glycolysis. This process is less efficient and produces lactic acid, which lowers the cytoplasmic pH, make it more acidic. To remove excess acid, the cell brings in sodium ions, but with less ATP available, the vital sodium-potassium pump fails, causing sodium, calcium, and water to rush into the cell, leading to swelling. Normal enzymes, proteins, and pumps in the cell work best at a pH near 7. Acidic conditions from glycolysis disturb these functions, making the damage worse. A major turning point towards Irreversible injury is the disruption of calcium balance inside the cell. Under normal conditions, calcium is carefully stored in the cytoplasm, endoplasmic reticulum, and mitochondria. Each area has special calcium pumps powered by ATP. During ischemia, calcium channels open and calcium floods into the cytoplasm, activating harmful enzymes like protein kinase C, endonucleases, phospholipases, and proteases, for example, calpins. Calpins worsen the situation by inactivating protein kinase C and damaging the calcium transport proteins. This leads to a calcium overload, especially in the mitochondria, which further damage the cell. If the cell cannot restore mitochondrial function, the swelling becomes irreversible and the cell will eventually die. Now we are going to discuss about membrane damage as a trigger for cell death. Free radical injury. Failure to restore mitochondrial function and repair damaged cell membranes is a key factor in irreversible cell injury. When oxidative phosphorylation is disrupted and mitochondria can no longer regulate calcium properly, the risk of cell death increases. Damaged membranes become more permeable, allowing calcium from outside the cell to enter. This calcium overload worsens mitochondrial damage, disrupts the cytoskeleton, and activates harmful enzymes like endonuclease, proteases, and phospholipases. Phospholipase A breaks down membrane phospholipids, further damaging both the cell and mitochondria, pushing the cell towards irreversible injury and death. Free radicals like reactive oxygen and nitrogen species are normally involved in cell signaling and defense against microbes. However, during conditions like ischemia, reperfusion injury, they can damage cell. These molecules have an unpaired electron, making them unstable and highly reactive. They attack the polyunsaturated fatty acids in cell membranes, starting a chain reaction that causes membrane breakdown. This process is called lipid peroxidation. Antioxidants such as superoxide dismutase, also abbreviated as SOD, catalase, glutathione peroxidase, and vitamin A, C, and E help protect cell by neutralizing free radicals and stopping the chain reaction. Students, now we are going to discuss about different forms of oncotic necrosis. Necrosis can be grouped based on how it appears under a microscope, which can help in diagnosis. This classification is influenced by the type of tissue affected, the cause of the injury, and how much time has passed since the cell died. The main types of necrosis include coagulative, caseous, liquefactive, and gangrenous necrosis. Now we are going to discuss about early hypoxic cell death. Coagulative form. Coagulative necrosis occurs when proteins inside the cells, especially in the cytoplasm, become denatured due to injury. This gives the cell a pink, glassy appearance under the microscope. It is commonly seen in early stages of hypoxia, ischemia, or exposure to toxins. The damage or resulting acidic environment not only denatures structural proteins but also inactivates lysosomal enzymes that would normally break down the cell. Because of this, the cell structure remains intact for some time, even though the nucleus shows signs of death like pycnosis, karyophyresis, or karyolysis. The term caseous comes from the Latin word for cheese, describing the soft, white, and crumbly appearance of this type of necrosis. It usually indicates an older lesion where the normal tissue structure is completely lost. Under the microscope, the affected area appears as a granular, pink, eosinophilic material with scattered blue, basophilic, nuclear debris, but individual cells can no longer be identified. This type of necrosis is commonly found in the center of granulomas, especially in infections like tuberculosis. Sometimes calcium deposits may form, adding to the granular texture. Now we are going to discuss about liquefactive type of tissue necrosis. Liquefactive necrosis involves the breakdown of cells, turning the affected tissue into a fluid mass. It commonly occurs in the brain or spinal cord, where the lack of supporting fibrous tissue and the presence of lipid-rich enzyme-filled cell leads to rapid tissue softening and liquefaction. Now we are going to discuss about overview of gangrenous necrosis. Gangrene is a form of tissue death that usually affects the outer parts of the body like limbs, ears, tail or lower parts of internal organs such as the lungs or mammary glands. It is mainly classified as dry or wet gangrene, which are distinct conditions. Wet gangrene occurs when the dead tissue becomes infected with bacteria. If these bacteria produce gas like Clostridium species, the condition is called gas gangrene. In the lungs, wet gangrene can develop as a complication of tissue breakdown caused by aspiration pneumonia. Now we are going to discuss about apoptosis, programmed cell death. Unlike oncotic necrosis, where cells swell and burst, apoptosis involves the shrinking and condensation of cells. It is programmed follow-up of cell death that plays a key role in embryonic development, maintaining tissue valence and natural reduction of organs or tissues when hormones or growth signals are lost. Apoptosis is tightly regulated and occurs in response to specific signals or cell damage. Apoptosis inducing triggers. Apoptosis or programmed cell death can be triggered by several signals. These include the binding of specific molecules like tumor necrosis factor to death receptors on the cell surface, cellular stress or injury caused by toxins or reactive oxygen species, lack of nutrients, withdrawal of growth factors or hormones, damage to DNA or attack by immune cells such as cytotoxic T cells and natural killer cells. Apoptosis can occur through two main pathways. The first is extrinsic pathway, activated when external signals like TNF bind to death receptor on the cell membrane. And the second is intrinsic pathway, initiated from within the cell, especially by mitochondrial signals in response to stress or DNA damage. Both pathways involve the activation of special enzymes called caspases. Caspases are a family of proteins that break down other proteins in a controlled way. Initiator caspases start the process. Caspase 8 is activated in the extrinsic pathway. Caspase 9 is triggered in the intrinsic pathway through a structure called the apoptosome. Caspase 2 is activated by the p53 protein in response to DNA damage. These initiator caspases then activate effector caspases like caspase 3, caspase 6 and caspase 7, which carry out the actual cell death process by breaking down key cellular components. Extrinsic death receptor initiated pathway of apoptosis. Extrinsic apoptosis is triggered when ligands bind to death receptors like first apoptosis signal receptor, also called CD95, tumor necrosis factor 1 or tumor necrosis factor related apoptosis inducing ligand receptor on the cell surface. This activates adapter proteins such as TNF receptors, associated death domain and FAS associated death domain, which recruit and activate caspase 8. Caspase 8 then activates executioner caspase to carry out cell death. It can also activate the mitochondrial pathway by cleaving BID. Proteins like Fly's inhibitory protein, also known as PHILIP, can block this process by preventing caspase 8 activation, while receptor interacting protein kinase 1, also known as RIPK1, can direct the cell towards survival, apoptosis or necrosis based on its modifications. Even cells that cannot initiate or propagate apoptotic signaling can still die. but do so via caspase-independent pathways of cell death, such as regulated necrosis. Intrinsic or mitochondrial pathway of apoptosis. The intrinsic pathway of apoptosis is activated by internal stress. Signals such as DNA damage, oxidative stress, or lack of survival signals. These stressors disrupt the balance between pro-apoptotic, which include BCL2 associated X protein and BCL2 antagonistic or killer, and anti-apoptotic B cell lymphoma 2 and B cell lymphoma extra large members of the BCL2 protein family, leading to permeabilization of the mitochondrial outer membrane. As a result, cytochrome C is released into the cytoplasm where it binds to APAF1 and ATP, forming the apoptosome. This complex activates initiator caspase 9, which then activates executioner caspase such as caspase 3, culminating in the orderly breakdown of cellular components. This pathway ensures that damaged or unneeded cells are removed in a controlled, non-inflammatory manner. This was all about today's topic. See you next time. Thank you.